Hi, in this video we're going to be looking at osmosis. There is more content, quizzes and past papers on the website, which is www.lfis.co.uk. Substances can move through membranes in three different ways. We have diffusion, osmosis and active transport. In this video we're going to be looking at osmosis. So what actually is osmosis? Osmosis is the net movement of water molecules through a partially permeable membrane from a dilute solution to a more concentrated solution. It happens when the total concentration of all dissolved substances inside and outside a cell are different. When this is the case, water will move across the membrane from a dilute solution to a more concentrated solution. Osmosis still follows the concentration rule from diffusion. The water moves from a place where it is in a high concentration, hence the dilute solution, to where there is a lower concentration of water, which is going to be a more concentrated solution. With the dilute solution and the more concentrated solution, think of it in terms of squash. If we had a very dilute squash, it would mean that there's very few squash particles relative to water, and this means that there's a very high concentration of water relative to the squash particles in a dilute solution. If we had a more concentrated squash, which we would get by pouring more squash into water, this would mean that there's a higher concentration of squash relative to water, and therefore a lower concentration of water relative to squash. Osmosis can only occur if the cell membrane is partially permeable. Partially permeable means that it lets through certain substances, such as water, and doesn't let through other substances, such as the solutes. Osmosis is important in moving water from cell to cell and from blood to tissue. It is important that the cells in the body are in a solution that has the correct concentration of solutes. If this is not the case, the cells can be damaged by osmosis and unable to perform their intended function. Let's have an example. Let's have a look at a red blood cell. A red blood cell is responsible for carrying oxygen around the body. If red blood cells are placed in water, water will move through the blood cell's membrane and into the blood cell. This results in the red blood cell swelling up and eventually bursting. However, if we place a red blood cell into a salty solution, the water will leave the red blood cell into the salty solution, and this results in the red blood cell shrinking. So let's have a look at an example. The example we're going to look at is whereby we place Viscin tubing with a sucrose solution inside a beaker of water. Viscin tubing has holes in it which allows water to pass through it, so we can say that it's permeable to water, but it does not allow larger molecules to pass through it, such as the sucrose, so it's impermeable to sucrose. Let's now have a look at a molecular diagram of this experiment taking place. Here we have a partially permeable membrane. The sucrose molecules are too large to pass through the partially permeable membrane, but the water molecules are small enough and are able to pass through the membrane in either direction. The water molecules are able to pass from outside the Viscin tubing to inside the Viscin tubing, and from inside the Viscin tubing to outside the Viscin tubing. The water molecules in the sucrose solution, which is inside the Viscin tubing, are attracted to the sucrose molecules. As the water molecules are attracted to the sucrose molecules, the kinetic energy of the water molecules reduces. This results in fewer water molecules moving from inside to outside the Viscin tubing. Whilst this is happening, water molecules are moving from outside to inside the Viscin tubing, which on the diagram is from left to right. The overall effect is that more water molecules move inside the Viscin tubing, which is the same as saying water molecules move from a more dilute solution to a less dilute solution. The more dilute solution is pure water, and this is on the outside of the Viscin tubing, which on this diagram is the left, and the less dilute solution is the sucrose solution, and this is inside the Viscin tubing, or on this diagram is on the right. But what influences the rate of osmosis? One factor that influences the rate of osmosis is temperature, whereby a greater temperature means a greater rate of osmosis. This is because a higher temperature gives the water particles more kinetic energy. This means that they move around faster and therefore cross the boundary quicker, which then means that the rate of osmosis increases. Another factor that influences the rate of osmosis is the concentration gradient. The lower the concentration of the solute within the solvent, the faster osmosis will occur. The rate of osmosis is also influenced by the surface area to volume ratio. The greater this ratio is, the faster the rate of osmosis. These three factors are similar to what influences the rate of diffusion.
The final factor that influences the rate of osmosis is completely different. This factor is the water potential. Water potential refers to how free the water molecules are to move. Pure water has the freest molecules and therefore the highest water potential. A more concentrated solution has a lower water potential. This is because there are more solutes for the water molecules to be attracted to. In the example that we had above, water is moving from a solution with a high water potential, which is the pure water, to a solution with a lower water potential, which is the sucrose solution. If we were to increase the concentration of sucrose in the sucrose solution, that would mean that the water potential of the sucrose solution would reduce further. This would increase the rate of osmosis. This brings us to the end of this video. For more content, quizzes and past papers, go onto the website, which is www.elevise.co.uk.